Welcome to NTI Week 4. Here is some more about viruses. I'm going to briefly cover the discovery of viruses without reading that textbook to you. So, viruses were discovered in the late 19th century. There was a whole lot of microbiology going on then. We were figuring out lots of things about pathogens. And there was this device called the Pasteur-Chamberlain filter that appeared to do a really good job of filtering out all kinds of bacteria. And it still does to this day. This scientist, Dmitry Ivanovsky, discovered, though, that when he took plant extract, ran it through the filter, and applied it to tobacco, that's what TMD stands for here, tobacco mosaic virus, that you could still get an infection of this disease, even though the extract had been run through this filter. This was quite puzzling because while we were just on board with the idea that germs cause disease, the germ theory of disease, now that here was a new problem where you had a disease that, where there shouldn't be any bacteria. So he suspected or he hypothesized that this was caused by some kind of a heretofore unknown tiny bacteria. A little bit later on, a scientist named Martinus Bejernik, I'm probably killing that pronunciation and I apologize. He hypothesized a little deeper and he kind of coined the term virus. And the actual credit for virology went to Louis Pasteur. He's generally credited as being the father of immunology, though Edward Jenner was doing work with variolation, which is a type of inoculation where you take a little bit of infected material and you stick it into an uninfected person. This was done with smallpox, and though he didn't invent it, Edward Jenner didn't, it comes from a very ancient Ottoman Empire practice. The idea being that if you were already healthy and you got just a very small amount of the pathogen, that your body could become immune and you would have be at less risk of dying. And it did bring the mortality rate down to about 3%. But again, this is variolation. This is actual live. This is pus, literally pus from a wound. From a sore that is stuck into you, right? Now, the mortality rate normally was about 30%. This was long before we had ways to try to keep people alive under extreme conditions like that on their bodies. But anyway, Edward Jenner, using work that was developed in the 1400s or earlier. Maybe someone can make that a project and let me know more details about it. Anyway, so these were viruses. Viruses, here are their basic characteristics, right? They are acellular, so they don't have organelles. They don't have they're not considered alive in the regular sense, right? They are, the fancy term for that is they are obligate intracellular parasites with host and cell type specificity. What that means is that they cannot replicate themselves without a host cell, and they are specialized for a certain kind of host cells, because the first thing a virus has to do is be able to attach. In order to attach, if you remember, everything in molecular biology is basically tiny, magnetic articulated Legos that are tiny. There are cell surface receptors for other things that viruses can exploit with little spikes on their capsids, on their outside coats. And they're able to basically fake a signal for something else. And in the case of viruses for animal cells, for eukaryotic cells in general, they trick the cell into bringing them inside. That's how they penetrate. Now, bacteria have their own... bacteria have their own types of viruses, which look something like this. Bacteriophage T4. Let's use this fancy new drawing tablet I have. All right. Sorry. Oh, there we are, right here. Yay. Oh, there we go. Okay, I'm going to have to practice with this quite a bit to figure this out. But yeah, the T4 bacteriophage, which I always thought was kind of funny with Resident Evil. But anyway, they have their own kind of viruses. And in bacteria, this virus has to attach, like all viruses have to do it first. And then it literally penetrates like a hypodermic, injecting its DNA into the cell. Now, I think I'm skipping around a little bit, but that's fine. Other viruses, like our coronavirus, which is will look kind of like this adenovirus, will use one of these spikes to pretend to be something else, and it will bind to the cell. It'll trick the cell into bringing the whole thing inside. So it penetrates completely, whereas the bacteriophages will, dock, they will attach and inject. The all of our viruses get engulfed, including like the HIV virus here, the retrovirus here. Okay, a little bit more about their structure, right? And I'll talk some about transmission as well, because that's important. I'm going the wrong way. Here we go. Oh, I definitely went the wrong way. All right, so viruses, characteristics. They have DNA or RNA, never both. And that little genome is packaged up in a capsid. So they sometimes are enveloped on top of that. So they'll have a little capsid, they all do, that will protect their DNA or RNA. And then sometimes they have an envelope on the outside of that. And in order for, they don't have the genes needed to reproduce on their own, so they need to signal their host cell they need to trick the host cell into making copies of them. Okay, so again, the virion, the mature virus particle that's able to infect, that is outside of a cell. It has a capsid. Got to start getting used to trying to use this. There we go. That's made of capsomeres, which are protein subunits. Remember, proteins, have jobs. And they make up the capsid. And then the categories, they can be naked. Now, if it is a naked virus, it means it only has its nucleic acid and the capsid. If it is enveloped, then it has a little bit of the cell membrane from its host that butted off with it. And, outward and away, some naked and enveloped, or spiked, right? They have their spikes. They can be in the envelope or they can be from the capsid. That's the point there. Here's the first view of the tobacco mosaic virus, which wasn't until around 1940. And we can view, we have fancier views. I'll show you here. The scanning electron micrograph, and here's a transmission electron micrograph. So the scanning electron microscope can give you detailed surfaces, which you then colorize, which is what's been done here. Whereas the transmission electron micrograph gives you cross sections, like cut through it. All right, now relative sizes, viruses are small. That's why that filter was able to let viruses right on through. There's the flu virus, a relatively large virus at 100 nanometers. And then, but it's smaller than mitochondria and bacteria, way smaller than red blood cells. The scale is, each one mark is times 10. And it's logarithmic. But again, it's bigger than proteins because it's, capsid is made of proteins, right? But they are tiny, is the point there. And before I talk about transmission, let's see, did I miss anything else? Nope. I'll move on. Skipping a lot of other stuff here. And hopefully you can't hear Quinn yelling at the Xbox in the background. Now, the other video covered the basic. Well, I'm still going to go over it again a little bit here. Or am I not? Yeah, I will. So the influenza virus is going to be like coronavirus. They're going to share the same kind of approach here. This is the basic lytic infection cycle. So, which I previewed a I think last week, the terms anyway. Okay. Now it's this way because the virus will attach, it will get engulfed by endocytosis, then the viral contents will be released. So the viral RNA will go to the nucleus to get the where it will be replicated by RNA polymerase. This is so sorry, I don't want to make this super complicated because it is, but overview: it gets in, it gets copied, it gets packaged, and then they get released. Now, in this particular scenario, the cell's not killed. The cell can continue producing mature virions, right, and releasing them. Often in the lytic life cycle, the cell will explode, releasing a ton all at once. Okay. But that's lytic. So here's an example of that in colorized scanning electron micrographs. These are all those little T4 bacteriophages attached. These are E. coli. And it literally blows up, see? Just blows up. Hijacks the resources, uses it for all it's worth, and then releases tons more bacteriophage. All right, we're not doing the clinical studies. Those are kind of neat. Isn't quite the class for that. So transmission. So there's the difference between droplet. This is droplet. And this is airborne over here. Wait, is it born? B-O-U-R? I don't know. Born. Airborne. So measles can easily go 6 feet because it can get spread by very, very fine particulate and hang in the air for hours. Ebola, and also this is true for coronavirus as well, hangs in droplets and doesn't go as far, right? Now, obviously, I talked about sizes. I talked about the Chamberlain-Pasteur, the Chamberlain filter, the Pasteur-Chamberlain filter, right? The masks, they can't stop the individual virus. That's pretty clear, right? N95, does a better job. But even the cloth masks will catch these big droplets. So the virus needs these droplets to get transmitted like that. So if you cough on the inside of your mask, you're catching those droplets. Now they could soak through and be exposed to the outside, right? But they're not going to be floating on the air as easily. Unless you really have explosive coughing fit and you're spreading virus like that. But by then you might be sick enough that you're not running around outside, right? Okay, so viruses and diseases. Let's see, that's transmission, which I'm going to come back to. Let's see if I have... Oh, here's the picture for this. So here's some of the variety of diseases that are caused by viruses in humans. We have the encephalitis, meningitis viruses. Measles can cause that. Now, infection of the brain, which is what encephalitis, meningitis is referring to, different parts of brain tissue, spinal cord there. There's a lot of viruses and bacteria that can cause that. But in this case, measles is particularly good at it, along with rabies. So we have our common colds, our rhinoviruses, parainfluenza virus, and RSV, which is not terribly different from SARS. But so SARS, several years ago, we had a concern with SARS. There was a lot of activity that took place to make it seem like it wasn't a big deal, but that's because it was handled. We didn't handle the situation well. But as you can see, there are other coronaviruses, and they've been around. But this is a SARS coronavirus. It's just worse than the ones that came previously. But yeah, a wide variety of diseases and a wide variety of systems. These are specific. So the ones that cause pneumonia tend to be have receptors that are best matched on the lung cells, right? Okay, let's see. Vaccine for... Does this one talk about vaccines? I'm going to come back to vaccines in its own presentation because it deserves a little more attention, right? All right, so transmission. Here's some of the terminology when we're talking about transmission. Direct contact is one way, and coronavirus definitely can be transmitted this way. Doorknobs are pretty notorious for this. Handles for things like even these. I should be using a pen for this. Pen tool. There we go. These handles. So you're turning them, if you're turning them off with your hands still, if you just washed your hands, whatever. But anyway, these are, direct contact with the pathogen. Here we're mediated by fomites, which are contaminated non-living objects. There we go. So there's also vectors. Vectors is something that can transmit the pathogen. They can be mechanical or biological. Here's how we tell the difference. Mechanical ones is like on the insect body. It lands in pathogen. The other stuff it lands on too. And then lands on your sandwich. It is not infected with it itself. It's literally just carrying that over to you. And then vector transmission biological is when, in this case, we're looking at a flea that was on a rat. So the pathogen was here and got carried to you through the flea into you. Like Lyme disease is supposed to be really bad this next summer, but if we're still social isolating and we're not going out as much, maybe Lyme disease won't be as bad, right? So when a disease is transmitted from an animal to a human, it's called a zoonosis, or they are plural zoonoses. Human to animals, reverse zoonosis. But yeah, I know we're still animals anyway, but... Okay, so there was the lytic cycle. There's another cycle called lysogenic. And in the lysogenic cycle, can I change ink colors on my... I wonder if I can. Ooh. So in the lysogenic cycle, so when it does this part, when the DNA goes into here, and instead of doing this, it stays in here. It integrates into the host chromosome. This is going to take practice, right? Maybe I should set it. Oh, hey, if I set it down and then write on it, let's see if that helps. Where it stays. Oops. Until it stops staying, until it something activates it. And this is the case in chickenpox. If you've had chickenpox, hopefully all you have been vaccinated for it. But if you've had it like me, I have this virus integrated into some nerve cells. And if I get under enough stress or sick enough, then on top of everything else, I might have that virus come back out. Yeah. Okay, so let's see. Moving forward, we'll be talking about immunity, but I think for this week I'm going to focus on the viruses themselves, and I need to give you some kind of a thing to go with this. But anyway, next week we'll look at the immune system in more detail, and from there we'll pull that back together with vaccines. But if you want a little preview here, oh look. I've watched some more of that show, Cells at Work, which is on Netflix. I recommend it. So I've matched some of the cells with their characters. And if I can find a way to deliver that to you, preferably in a legal way, I will try to do that. All right. Anyway, that'll be next time. We'll go over innate immunity. And I guess for now, this is good, along with whatever assignment that I package with this. All right. Again, you know how to contact me. Contact me for whatever this stuff you need help with. I'm online every day on Google, on that Google Meet page. So I'll talk to you later. All right, bye.